Today's topic is bacterial chemotaxis. So, what is chemotaxis? Chemotaxis is the directional movement of an organism or a living motile cell in response to certain diffusible chemicals in the environment. Here, diffusible means the chemical which can penetrate the cell membrane by osmosis. Now, chemotaxis are of two types, positive chemotaxis and negative chemotaxis. When the movement is towards a higher concentration of diffusible substance, then it is positive, and if it is towards the lower concentration gradient, then it is negative. Now, we will explain this one by Bazarin's experiment. For this purpose, we have to take 3 slides and we have to put aerobic bacteria, anaerobic bacteria and microaerophilic bacteria in these slides respectively, and then cover by cover slips. Observation. We will observe that the first case, the strongly aerobic bacteria collected in large numbers at the margin of the cover slip. And in case of the middle one, that is anaerobic bacteria, they move to the center of the cover slip. And in the last case, that is microaerophilic bacteria, they will be collected at some distance from the margin of the cover slip. Here the stimulus is oxygen. Now we can say that bacteria can regulate their flagellar movement in response to the stimulus, either positively or negatively. Here the stimulus is oxygen. They exhibit a directional movement towards a concentration gradient of the chemical stimulus. If the medium is homogeneous, that is without any concentration gradient, then bacteria show non-directional movement. Here we have observed that in the first case, bacteria moved towards the margin of the cover slip because the bacteria are aerobic. In the second one, they are towards the center because they are anaerobic. And in third case, they are not far away from the margin because they are microaerophilic in nature. Now we will discuss the mechanism of bacterial chemotaxis. To perform chemotaxis, bacteria utilize a sensory system having chemoreceptors and transducers. At first, the chemoreceptor proteins located at the periplasm of gram-negative bacteria receive the chemical stimulus on the cell surface. Next, chemoreceptors transmit a signal to the transducer proteins which are located in the cytoplasmic membrane. In the next phase, the transducers transmit that signal to the flagellar motor through small cytoplasmic proteins. At least 2 cytoplasmic proteins appear to be involved. And in fourth case, that is step, the direction of rotation of the flagellar motor is controlled by methylation and demethylation of the transducer proteins. And methylation of transducer proteins activate the cytoplasmic protein by phosphorylation, that is with the expense of ATP.